As we were discussing in the last class about the water and its property, so today we are going to look at like some of the bonds that might be asked during your MCQ as well as MSQ preparation at IIT JAM or GATE. So when we talk about the non-covalent interactions for the water or like for any particular molecule, so there are hydrogen bonds which we just discussed. If you want, you can watch the previous lecture. So these are basically special dipole-dipole interactions. Okay, special dipole-dipole interaction. So there is an electronegative atom such as oxygen or nitrogen, fluorine, etc. Okay, to this if any hydrogen is attached, so this particular hydrogen can make a hydrogen bond with any other electronegative atom such as O, N or F. Okay, so this is the very important bond in case of protein folding, in case of DNA, in case of even small molecules. Okay, so that's why this is so important. Now the second thing is van der Waals interactions. Van der Waals interaction. So these are basically happen in non-polar molecules, and these are basically hydrophobic interactions in simple words. So for example, CH3, CH2, CH3, and there's another molecule called CH3, CH2 and CH3. So the interaction between these two molecules is called van der Waals. It's as simple as that. You do not need to remember like much of it. So in some of the cases what happens is if there is a certain electronegative or electropositive atom attached to like one of these, so for example in case of H, so if there is an O negative, okay, so there will be a induced dipole. Okay, so this particular portion becomes negative, partial negative, and by the convention this becomes partially positive, so this has a dipole now. Okay, and this dipole can induce a dipole into another molecule also, which is also called dipole induced dipole. So this is another form of like interaction, a bond. And the third that we already know are the ionic bonds. Ionic bonds. So here we have a electropositive atom and we have electronegative atom. So when these make a bond, that is called ionic bond. Okay, it's as simple as that. So now let's learn about the hydrophobic interaction in case of water. Hydrophobic interaction. It is also known as hydrophobic effect. So it actually talks about relation between water and a hydrophobic substance. So what is the most common hydrophobic substance? Just think. So for example, if I have this particular mug, let's suppose this is water, right? So what is the most common form of like hydrophobic substance we have? So we have oil. Okay, so let's suppose this is oil. So what happens is this oil will repel the water, right? And this repelling is happening because there is no hydrogen bonds that can be formed. Hydrogen bonds that can be formed, as we are going to study the basic structure of lipid, but it's something like this. Okay, so there are carbon 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 chain and there is like carboxylic in the one end, right? So due to these carbon carbon chain, so there is no direct interaction that can happen with a water molecule. As we have studied, we need a electronegative atom so that it interacts with this H2O and make a hydrogen bond, right? But in case of water, no such possibility occurs. Okay, so in this scenario what happens is, so again I'm drawing the water. So for example, this is a water molecule, right? No, the surrounding is the water molecule and this is the lipid or the fat, okay, or oil, whatever you want to say, right? So what happens is, so the water molecule are going to constrict itself so that it repels and excludes the oil or the fat molecule. So these type of interaction is called hydrophobic interaction. where water is actually repelled. Okay, so in all of this, so now understand the thermodynamics. So if this is a water, right, so each water molecule can move freely around this particular area, right? So the moment you are going to put the lipid or the fat in there, so water molecule are constructing itself around the lipid. So now these water molecule cannot move freely. Now due to this, what happens is, so there is a loss of entropy, loss of entropy, right? Although there is a loss of entropy, now what happens is, if I put like lots and lots of like these lipid molecule, so every lipid will be covered by like these kind of water molecule. Now due to which what happens is, so there is a much higher loss of this entropy. Now to reduce that, what water molecule does is, they aggregate every of this into one particular space so that they can increase the entropy of the system. So they'll merge all of these lipids together, and they overall creates the entropy of the system higher. Okay, so they'll simply merge. So now the fewer water molecule will be needed to surround each of this particular lipid, and due to which the entropy of system gets increased. Entropy of system gets increased. I hope you get the idea. If not, let us understand again. Okay, so understand. So this is a, right. So this is so this is water, right? So if in this water, if I add some sort of lipid molecule, right? So just suppose this is lipid, this is lipid, this is lipid. So now due to the convention, so these water molecule will get constructed because they have to repel this molecule. Molecule in the sense they have to repel the oil or the fat, right? Now the surface area of water per lipid is much higher. Okay, so now just suppose if all of this, so if all of these are like merged together, so now the overall water molecule that will be needed for constructing these lipid will be much lower. Okay, so thus hydrophobic interaction increases the entropy of the system. And why it is happening? Because fewer water molecules are needed to surround the aggregate than to hydrate each other. Okay, although we are losing entropy of the system for the very first case, because here water can move freely, but in this case there is some sort of constriction, so there is a decrease in entropy. But again, due to the hydrophobic interaction, so there is an increase of this entropy by aggregating all of these lipids together to form this particular structure. I hope you get it. Okay, so now take the definition. Exclusion of non-polar substances from aqueous solution is hydrophobic effect, right? And this hydrophobic is again important in protein folding, DNA folding, and in medicine. And I'll give you example why. Okay, although we'll understand later. So what happens is, so just suppose we have a protein molecule, right? So let's suppose this is a protein molecule now, which is water soluble. So it will easily solubilize in the water and will have no effect. Now just suppose there is a particular protein which has very hydrophobic groups that we are going to study in upcoming lectures. Okay, but I think you understand what are the hydrophobic group, which basically repels the water. So in this case what happens is, so if protein is something like this, a linear chain, okay, a linear chain. So in this case what happens is it is going to constrict itself to a particular like shape so that it excludes the water molecule, and it will form a structure like this that I've just made. Okay, so it will make a structure like this to exclude the water molecule. So this has direct role in protein folding. I hope you get it right now. Okay, so we have already exceeded the 10-minute mark, so we are going to continue in the next lecture. So now we'll start with the acid bases and buffer in the next lecture. So if you got confused, so watch this lecture again, and I hope you'll understand it. It is a very important and very initial lecture for the biochemistry, because if you understand this, everything is going to be easy. We are studying water because every biomolecule is in the water, and why water is essential, that's why we are studying. Okay, and take one more thing. So here I forgot to mention delta G is equal to delta H minus T delta S, and all of this process is entropy driven, okay, which I have like discussed. Thank you, and bye bye, and subscribe.